So we just have Unit 10 left to go over the practice questions. Don't forget you still have to study the vocabulary on your own and make your cheat sheet. You can't bring this study guide into the final with you. And the expectations are on the front page of the study guide about your cheat sheet. Okay, number 1 for Unit 10. In DNA, cytosine pairs with guanine. You could just write G. See, I said you may use letters. You don't have to write the entire word. So you could just say C pairs with G. That's fine. In DNA, adenine pairs with thymine. So C and G go together, and A and T go together. In DNA, thymine pairs with maybe adenine. So that's another thing I would suggest writing on your cheat sheet. The rules for base pairing: A with T for DNA, and C with G for DNA. In RNA, A pairs with U, uracil, and G still pairs with cytosine. Uracil pairs with adenine. So it's A, and for RNA, it's A goes with U goes with A, and then C and G go together. Okay? Those should be easy points for you guys. You've seen this 6 times already, this image. Okay, the image to the left represents and you have a word bank, DNA molecule. DNA molecules are made up of nucleotides. There are 3 parts to each of these building blocks. So each building block has a sugar. I can just write sugar, or you can write deoxyribose sugar specifically, since it's DNA. A nitrogenous base, or just base is fine, and a phosphate group. What type of macromolecule is DNA? We've already had a question like this, actually. What does the Na stand for? Deoxyribonucleic acid. So it is a nucleic acid. I actually messed up on question 13, and I think it might be this way on your final. So transcribe this DNA template strand into mRNA. Well, this is not DNA because I put U's already. So you can just use your simple RNA pairing rules here. A goes with U goes with A, G goes with C. So I'm just writing down here. I can scoot it over. Oops, I didn't mean to hit that. So you can just see it. Okay, I'm just putting the complement. A, C goes with G, U goes with A, G goes with C. There. So this is going to be, if this is the template strand, this will be the mRNA from that template strand. Obviously, I made that mistake of putting U's instead of T's. But that actually kind of makes that question easier for you guys. 14, I forgot to delete this. Go away. Using the mRNA codon chart to the right, what would the 3 amino acids of this, another typo, fantastic, of this, oh no, that's correct. What would the 3 amino acids be if this was the mRNA strand? Okay, so AAU, gotta look that up in the codon chart. First 3 letters, AAU, A, first letters on the side, so I look up A, then U is the first column, so AU, I'm sorry, it's AA. So in this box, AAU is ASN. See right there? So I'm going to write that right here, ASN. Okay, ACG. A, second column is C, G would be THR. And then UCG. You can like circle these or like just write them, you know, a little tricky to not get your codons messed up. But so separate out the codons is a good way to make sure you're not making a mistake here. UCG, U, then C, G would be serine. Serine. Go. That question's done. And okay, can a cell survive without proteins? Simple yes or no? No. Why is it important that cells can make proteins? Proteins are used for most functions. A cell. Which organelle makes proteins? Ribosomes. And that's it.